Buenos dias. Thank you, Victor, for the presentation, for this nice introduction. And first of all, to begin with, so first, thank you, Steve, and thank you, Victor, for, I'm audible. Okay, so thank you for inviting, and thank you for arranging such a wonderful presentations and the symposium. So here, as Sudhakar, thank you for this nice view of the solubility enhancement and solubility technologies and the salt formations. So it was really wonderful presentation, Sudhakar. So here, mostly I will discuss about the next role, like the technology that will. So here, my presentations will be mostly focusing on the solubility enhancement technology. And then how it will be useful, like the integration of solubility technology, like for example, solid dispersion technology, in which mostly I will discuss about the hot mill extrusion and the 3D printing technology. But how important that the integration or the use of the solid technology, like solid characterization technology is useful in order to develop the robust formulation. So we'll see how it works together. So during this presentation in next half an hour, we'll go through the solubility enhancement approaches. In that, we'll discuss mostly about the solubility enhancement by solid dispersion, like hot mill extrusion and 3D printing, in which it's been now almost 18 years I'm working in these three technologies. Then also we'll discuss about the 3D printing and then for how we can select the formulation. There are different technologies that how we can identify, like it is known as the pre-formulation studies, and there are some commonly used excipients as well as some plasticizer in order to make the process more robust. And then there are some case studies that I will discuss at the end of this presentation. So Sudhakar is already informed that there are, and I think most of us now are aware that there are 4 BCS class, class 1, class 2, and class 3, and class 4. But here my major focus is on the BCS class 2 and class 4, because these are the drugs which are almost 40 to 60% when anyone start with the synthesis or the new molecules. So mostly they belong to either class 2 or some molecules are class 4. So this composition is almost 40 to 60%. And that's a major problem with the formulation scientist. Because solubility, if there is no solubility, there will be no dissolution. And there is no dissolution, there will be no absorption. So it means there is no therapeutic indication. And so how we deal with this problem? So we'll go through it. So first one is, so here, BCS class 1, there is no worry because mostly they are soluble, so you don't need to worry, they are highly absorbed. But when it comes to the BCS class 2, they have low solubility but high permeability. But because of the low solubility, they are not getting absorbed. So dissolution is the major problem for the absorption. So we need to enhance the solubility, and by hot mill extrusion or by nanoparticulate drug system or micronizations, we can increase the solubility. The next one is the class 4 drugs, which is having the low permeability and the low solubility. And because of that, it gets poorly absorbed, but some molecules, case by case, it changes. So now coming to the technologies, what are the technologies that we can use for the solubility enhancement? As in the earlier presentation, we already heard that the salts is the major source for the solubility enhancement. But in addition to that, there are different technology as well that we can use for the solubility enhancement approaches. First one is the co-solvent. For example, the drug which is Prograf, which is already in the market, is developed by using the co-solvent technology. Then salt, diclofenac, there are various salts, hydrochloride, phosphate, or there are zinc sulfate. So you can use the salts and you can get the solubility enhancement, also the good permeability. Next is the surfactant that you can use different surfactants, for example, sodium lauryl sulfate, or there are different twin 80s. It depends on the characterizations of the drug molecules and the compatibility. And based on that, you can decide which surfactant you want to use. There are cyclodextrins, like hydropropylbutacyclodextrins that you can use. Then most common method that generally go for and the easiest method that go for the particle size reduction, maybe in micronization of the API, or the size reduction by nanomilling. So for example, Imane is based on the nanomilling technology, and they are spraying it on the pellets. So this is one of the particle size reduction example. Polymorphs, you can change the polymorph, and you can select which polymorph is stable. Then come to the lipid-based drug system, where is the, like Neurol and Agenerase is one of the classic example by using the lipid, which is developed by lipid-based system, co-crystals. But here my major focus will be on the amorphous solid dispersions. And as there are a number of molecules which is already developed by hot melt extrusions or solid dispersion by spray drying or different technologies. And in this case, when we use for the hot melt, when you go for the solid dispersion technology, the most common technology among all the approaches which majorly pharmaceutical scientists use is the hot melt extrusion, because they are getting more and more interest into the formulation because of their easier usage and different advantages. So how it will be useful and how the solid characterization plays an important role when you develop the solid formulation or the solid characterizations. So first one is, when you go for the solubility enhancement by using this kind of technology, so first one question is whether it is miscible, because you need to identify the polymer which you are using along with the drug. It should have the solubility with the polymer and the drug. So the drug carriers miscibility is an important role. In that case, you can use this kind of technology like differential DSC, and the XRD is a major characterization technology or tools which plays a major role in order to identify the drug carrier miscibility. Then drug carrier interactions, whether you are having the compatibility issue with the polymer which you are selecting, so you need to go for NMR or IR. Then physical structure that you are developing, whether it is in the crystalline form or into the amorphous form, you need to identify. So you can do the different technologies like XRD, DSC, and you can identify. So I will go and also the stability. So the next slide, I will focus on this more majorly. So when you go for the hot melt extrusion, so there are 3 chances that you can develop the formulation based on the polymer and the drug and the thermodynamic. If your drug is amorphous in the crystalline, and if your polymer is amorphous, so there may be a chance that you will get the almost stable formulation. So in that case, the TG means the glass transition temperature and the melting point will be the same. So in that case, the formulation will be stable and you will get the amorphous or solid crystalline suspension. If it is a solid glassy suspension, in the second case, the drug will be in the amorphous form and the polymer will be in the amorphous form. There are many polymers, they are amorphous form as well, and then after the melt extrusion, you will get but this mostly it's unstable. So in order to get the mostly the stable formulation, it is always recommended to go for the amorphous and the molecular dissolved. So in that case, what happened? Your polymer, the melted polymer will get work as a solvent, and your drug will get completely dissolved into that, and you will get a complete clear solution. So that is known as the most stable formulations for the hot melt extrusion. Now, why we are using the hot melt extrusion? So there are a number of applications and number of problems, and which is the single solution for the different problems. The first and the biggest problem that the pharmaceutical industry faces is the low and poor viability. So how hot melt extrusion helps in this? So when you use the hot melt extrusion, so it will prepare our solid dispersions, and because of the solid dispersion, the polymer and the drug, because of the polymer we use in the formulation, they convert the drug into the amorphous form, and it makes more soluble. So that's the first advantage of this hot melt extrusion. The second one is there are a number of APIs which are unstable. You can't use the water. So in that case, hot melt extrusion is a very good technology. You can use the melt extrusion, melting the polymer at maybe 60 degrees Celsius or 70 degrees Celsius, and no need to use the water. So that is the second thing that you can do. So no hydrolytic stress and no drying. Third one is this technology, because I use this technology for developing the modified release formulation also. Like I developed the metformin modified release formulation by using some like HPMC. So this is a good technology in case if you want to develop a modified release formulation. Third one is that if they have some problem in the stomach, so you can use this technology and you can cover this with some enteric polymers, and you can protect the drug from the stomach. And if the drug is having the bad taste or some poor taste, you can convert this into the better palatable drug formulation. So now coming to the design of the hot melt extrusion. So mostly, how many of you are aware of this hot melt extruder? Okay. So here, so I think most of them know that hot melt extruder. So I will explain it. So this is the hopper. It's difficult, little bit difficult different equipment than the conventional formulation that we use, like compressions or granulation. But here we can use it for the granulation. So here they have the hopper from where you can feed the drug, and polymer combinations. But there is another option. If you can put the hopper from here, there are different options that you can put the hopper from here. And from one hopper, you can put the drug, and from one hopper, you can put the polymer. But here, I put the one hopper. Here, you can put the mixture of drug and the polymer. In case if it's required, you can use the plasticizer based on your applications. Like in case sometime that we need to reduce the polymer processing temperature, or you need good solubility, so sometime we use the plasticizer. So after the addition of this, there are the screws. So these are the screws. You can change the screws as per your requirement, that if you need the high stress, so you can change it. Like with the 60 and 90, you will get the more shear pressure. So with the 30 is a less. With the 90, it will give the more shear pressures, and it will enhance the solubility more. So, and this rotate with this machine. And here it is like that. Here from the temperature, you can start from the 30 to 120 degrees Celsius or 200. It depends on your requirement, that what temperature you need for the processing. So it will rotate. There are a number of speeds that you can change, and then there are different zones. For example, here, so here after feeding, this screw will rotate. So this is like the feeding zone. Then this is the mixing zone where the polymer will get mixed together. And then afterwards, it's a mixing. This is the venting zone, because there are generation of some gases, and you need to take it out. So we need to keep some port. And then the metering zone. Here you can use the different die based on your requirement. Either you can use the screws or like noodle shape or any granules. So here I will go to the dosing zone. So here as I informed earlier, so here you can put the dosing. And the objective of this zone is to convey the material from hopper to the barrel. Next zone is the metering zone. In this zone, the polymer will get the mix, and also it will get conveyed to the other next melting zone. So in this case, here the temperature is ranging from 40 to 100 or 120 degrees Celsius, and most of the mixing and the melting is happening in this zone. Afterwards, when there is a generation of some gases, or there are some volatile materials in the formulations, that can be removed through this venting zone. So here is a venting zone, and all the generated gases, or if there are some volatile solvents, that can be evaporated from this. Once you have, here I put the extrudes like a noodle-shaped die. So here you will get the noodle-shaped die. They are noodles, then it will be crushed, or it will be pelletized by using some crushing machine or by granulator, and then you will get the granules which are free-flowing. Then you can convert it into the tablet dosage form, or either you can fill it with the capsule by using some other excipients like lactose or some other excipients. So now, how many of you I have a question for the audience. How many of you has taken the printout of your document in home or at home, at office? I think most of them, everyone. Then my next question is to all of you. How many of you has took the printout of the tablets? No one. But I assume, and I must say that by 2030, I think you can take the printout of the tablets at your home. I'm not joking, it's true, it's possible. So how it is possible that how we can take the tablets printouts? So here, the new technology where we use the hot melt, there are different technologies that we can use for 3D printing. But here, as I'm focusing on hot melt extrusion, so I'm connecting 3D printing with the hot melt extrusion. So what we do in the 3D printing, we develop a like soft copy of the tablets by CAD, and then we give the command to our printer, and it will develop the tablet, whichever the shape you want. And these are this technology mostly we are using for the personalized medications, or modifying the dose as per the patient requirements, especially for the epilepsy, because I was working for one of the applications for developing the 3D printing tablets for CNS disease. So here we'll go how the 3D printing works and how it is useful. So here also the 3D printing technology is also useful for the solubility enhancement, but in addition to that, it's also useful for the personalized dosage design. So here you can design the drug. If some patient, for example, levetiracetam, it doses from 500 milligram to 1 gram. But everyone, so it can't make a 300 gram. For patients, like for kids, maybe it requires 100 milligrams. So you can print the tablets for 100 milligram. That is the breakthrough that we can do this with this. So I will skip this one. Now, what we do with the 3D printing technology, so here we use the fuse deposition modeling. In this, what do you do? The polymer which we are selected, we are mixing with the drug, and then we are melting it, and then that we are using as a ink or as a printing material for the solid oral tablets. So here I will show you one example how we can print the tablets. So this is for the theophylline tablets, which will be developed by using the 3D printing technology. The video will not work. Okay, so no issue. So I think, yeah, afterwards I will share these presentations so you can see the video at home, so not a problem. So here actually we are developing the printout where we can feed the ink. Instead of ink, we are using the solution, the melted solution of the polymer, maybe like polyvinyl pyrrolidone along with your drug, for example, levetiracetam. And then it will print with the nozzle, and then you can form tablets. Like it will be 3D printed tablets. And based on that, if you need 500 milligram of tablets, you can give a command of 100 milligram. But of course, this is the prescribed medications that you will get it from the pharmacy up to the prescription of the doctors. So this is one of the technology that I want to show you. Then now, how to develop this hot metal extrusion? So of course, when you start working with the hot metal extrusion or 3D printing technology, so there are some pre-formation studies which are important. So how to select the polymer, how to select the processing temperature, what are the screw speed. So based on that, there are different pre-formation studies. The first one is the solubility parameters. So if your drug is water insoluble, BCS Class 2 and Class 4 drugs, and we want to use it for solubility enhancement. So the polymer selection is very important. So how to select this polymer? The first and very important thing is the solubility parameter. Here, the drug which you are using has to be of very high solubility, or it should have a very good miscibility with the polymer which you are choosing. In case the drug solubility is drug and the polymer solubility is less than 5, then the drug and polymer are not considered to be miscible. So the solubility parameters, which is developed by using the Hoffer and the Van Kreuling methods, they must have less than 5. So if they have less than 5, that it means the drug solubility of this polymer and the drug is very good. The second is the differential scanning calorimetry. I think most of you are aware that we use the DSC in order to understand the melting point and the glass transition temperature of the drug and the polymer. This is very important to understand that whether the polymer which you are using are stable at high temperature, and also the drug is not degraded at high temperature. So DSC is an important tool. Then, of course, if you want to see that the drug is in the phase, which phase is the drug, whether it is in the molecular dissolve, and we are getting the single TG, so DSC is an important tool in order to determine this kind of solid dispersion. And also, it will predict the miscibility of the drug with the polymer. In addition to that, what are the percentage of drug that you want to use? So in that case, you need to use the film casting process. So use the one solvent in which the drug and the polymer is getting dissolved. In that case, you need to use the different percentage of drug and solvents and try to make a film cast on the slides, and then dry it. And after drying, if it is getting precipitated, so it means that the drug combinations of that percentage is not suitable. So you need to find out which is not crystallizing, and then find out the percentage drug loading that you can use. So it should be transparent. Then the degradation temperature, that generally we use the TGA in order to find that you need to melt it at 200 degrees Celsius, 250, and if you see that 250 degrees Celsius, the temperature, the polymer and the excipients or the drugs are not getting, or they are getting degraded, so then you need to use the processing temperature less than, always 20 degrees Celsius less than your maximum degradation temperature. Then melt viscosity, because if you are using highly viscous polymers, like there are a number of polymers, for example, HMC to netium, but when you melt these polymers, their viscosity is too high, and then the processability will be the biggest issue. So in that case, the selection of the melt viscosity is very important, so that the screw will be not stuck, and also the polymer which you are using, it should not give like a viscous solution. So in that case, the melt viscosity is very useful. For example, shear thinning, dilacy, and thixotropy, and also the melting behavior, and also to avoid some mixing problem. Here are some compounds or the commonly used polymers. Generally, they are recommended for the hot melt extrusion and the 3D printing. For example, polyethylene glycol, HPC, Clucel, but there are different grades, so you need to select the grade, proper grade, because as I said, the melt viscosity is important. So higher viscosity polymers are not recommended for hot melt extrusion. Then PVP, and copolymers of ethylene oxide and propylene oxide, like Lutrol F27 and Lutrol F68, they are nowadays quite popular because of their simplicity and easy of use. So you have a polymer, but then why you use the plasticizer? Because sometimes what happens, the processing temperature that you are using, for example, HPC, and you need 200 degrees Celsius temperature. And if you want to reduce the processing temperature, so instead of that, you can combine the plasticizer along with the HPMC, maybe 10% like TPGS or vitamin E TPGS is the common solvent generally you use. So with this 10% or 15%, you can reduce the processing temperature, or in other case, we can increase the solubility maybe 10 times or 15 times by using this surfactant. So like for example, if HPC is using, alone is giving the processing temperature of 200 degrees Celsius with using the ETPS, so maybe processing temperature will be around 150 to 160, and the solubility enhancement maybe 30 or 40 times than the single molecule. So now, once you have the formulations, how you develop, so how you know that your formulation is better, whether you have achieved your objectives. So there are different technologies that we generally use, and here the solid state characterization technology is a very important tool, plays a very important role. For example, differential scaling calorimetry. If you have a quantity, if you want to do a quantitative detection for the transition like TG and TM, it is very important tools to go for the differential calorimetry. Also, the excipients which you have choose, maybe you use the TPGS or Lutrol or polyvinyl pyridine. So whether they have the compatibility, so you need to go to the drug to study the drug excipient compatibility with the DSC. Then their next tool is the DSC, the TGA, in which this is the screening tool. This is a screening tool where you can check the thermostability of the drug, the polymers, as well as the plasticizers. The XRD, that whether your formulation is really amorphous or still it is in a crystalline form. If it is an amorphous form, what are the amorphous formations? Like whether it is 100% or there are still drug which is not crystalline in the form, and there may be stability issues. So you can determine it with the X-ray diffractometry, and it will give the good sensitivity even the less than 10%. That IR in order to differentiate the polymorphic form, and of course the different peaks which are sensitive to the changes in the crystallinity. And finally, the dissolutions. If there is a real solubility enhancement, you can check it with the solubility or the in vitro dissolution. And the crystallinity of the material you can check with the NMR. Microscopy in order to see the drugs are into the crystalline form or they are into the amorphous form, or what kind of solutions they are. And of course, finally, the strength of the formulations that you can do with the mechanical analysis. I use this technology, hot melt extrusion, by using one anticoagulant drug molecules. The problem with that drug was it was like with the 10 and 15 milligram, with the 5 and 10 milligram, the drug was having no solubility issues, and there was no food-dependent viability. But when we change the strength from 15 to 20 milligram, as we go for the higher strength, the solubility was dependent on the food. So, and that formulation was showing the food-dependent viability. So in that case, when we go for the food, so the viability was 30 to 40% higher, but without food, it was less. So what I did, so because of this, the drug was the BCS Class 2 drug. So in order to avoid such kind of problems, I took this drug, I mixed, of course, with these pre-formation studies, I identified the TGA And of course, with these different polymers, I selected different polymers like PVP, polyethylene glycol, HPMC, and I found that PVP is the best polymer for that. And based on my pre-formation studies, I selected the PVP, and the percentage was approximately 15 to 20%, the drug loading. And then I went for the study, and then at around 150 degrees Celsius, I extruded the polymer drug combinations, and then I found that drug solubility is almost 30 to 40% higher, 30% higher. And when we go for the clinical study, so we found that our formulations, in the in vivo clinical study, we performed the bioequivalence studies. So our formulations was better or the show improved viability, and there was no food dependency. So either you go for the fasting or with the food, it is showing the food the same viability. Now, what are the next future of this technology? So, of course, we know that it's still there. We are lacking behind some like polymers. There are no novel, some polymers are coming up, but still we are not having a robust polymer which can be useful for hot melt extrusion and 3DP. So, number of companies like Merck, BASF, they are working on the development of the novel formulations in order to use these excipients for the hot melt extrusion and to get rid of this solubility issues in order to get the better absorption. Then there are identification of vehicles or excipients that may retard or prevent the crystallization, because even after the development of the formation, there are few polymers which can be converted. After 3 months, they will show that the formation is not amorphous, it is crystalline, and the solubility gets getting retarded. So there are some issues. So we are working, or most of the companies, innovative companies, they're working on identification of the vehicles or the excipients who can prevent the crystallization. Then, of course, we are in the next target, in coming years, we are working on the integration with personalized medicines with the number of doses form in order to get the dose as per the patient requirement. And of course, now the complex formulations are coming, like mostly the formulation which are now getting off patents, they are into the complex formulation. Mostly they are solubility issues, or some they want to convert to the modified release formulation. So developing the controlled release and the complex formulation will be a future. And what about the solid state characterization perspective? So we are now focusing mostly onto the synchrotron-based techniques, advanced spectroscopy in case like NMR and Raman, and also the integration, because now we are also moving toward the continuous manufacturing by hot melt extrusion or different technologies. So in that case, the inline analysis or the inline solid state characterization plays an important role. So like the PAT tools, near infrared or Raman spectroscopy will play an important role. So inline characterizations tools in the manufacturing process and the development will play an important role. Now we are also moving towards the AI and the machine learning. So the use of the AI and machine learning will be an integrations or will be the useful tools for our coming years. And the next one is the real-time tracking, because if you are working on the continuous manufacturing, so what will be the real tracking? Like in order to go for the batch analysis, you can do the inline analysis and find out what is the assay, what is the stability, so you can do this during the real-time tracking. And of course, the last one is the emphasis on the stability, because you are developing the formation, so it should have a stable, because they have some shelf life, so 2 years or 3 years. So the stability and the compatibility analysis is an important rules, especially for the solid state characterization. So this was all about the hot melt extrusion, solid state characterization, and 3D printing. If you have any questions, I'm open for it. Thank you. Thank you very much for listening to me.
Okay, questions? Anthony.
Hi, thank you. Thanks for the interesting talk. Another popular or common technology for preparing solid dispersions or amorphous solid dispersions is spray drying. What advantages does hot melt extrusion offer over this technique?
That's a good question, actually. So when I developed the process, so I was working as a solid expert, solid dispersion expert. So I tried this spray drying technology, but and I compare the same product with the spray drying and with the hot melt extrusion. The first advantage is the solvent, because for spray drying, you need to use the solvent. And this HME technology is solvent-free. So of course, you will save the cost. The second one is that the processing parameter, the scale-up is very difficult for the spray drying, But in case of hot melt extrusion, there are designs. The screw, you need to change the screw design. And of course, you can go for the easily to the higher scale. Third one is the processing time. So for spray drying, for example, 10 kg batch, it takes longer time. But for example, hot melt extrusion, it will go for the 2 hours or 3 hours, based on, of course, your formulation optimization. So, and of course, the cost. So these are the few differences. So when I compare, for example, acidipine I was working with, so I developed the same formulations with the spray drying. It's just to compare because I'm a scientist, so I just want to compare with both the process. So when I compare the acidipine development with hot melt extrusion as well as the spray drying, I found the hot melt extrusion was more faster with the less cost. So for spray drying for 10 kg batch, it was taking around 9 to 10 hours. But for the same product with 10 kg, we developed with the 2 kgs. And of course, the particle size which you get after the spray drying was not easy to flow. The flowability was the biggest issue. But in this case, this is not an issue. So, and of course, the solubility and the stability. So that is also with the hot melt extrusion, the solubility was better than compared to the solid dispersion, which I get from the spray drying. So that's the reason I moved from spray drying to the, of course, there are some issues with the hot melt extrusion also, but I think it's better to use the solid dispersion by hot melt extrusion because it's easy, more robust, and you will get the better solubility enhancement.
Thank you very much. Any other question in the room?
Hi, very good presentation. I have actually 2 questions. One is related to, up to your experience on HME right now, what is the lowest temperature you can actually effectively process a formulation, generate a formulation by HME? As you know, the main issue is thermal, you cannot handle or very difficult to handle thermal unstable drugs. And of course, it's always a matter of time, temperature versus time of processing. So what is your experience in that? Do you have a sort of criteria to determine which is the lowest limit or in which you can safely handle a molecule and so forth?
I think there is no limit. Because when I was a part of Dr. Reddy's Center of Excellence team, I developed the metformin modified release formulation. Mm-hmm. And in that case, I didn't use the temperature. But that technology I use for the continuous manufacturing. So what the idea is, because from last 10 years, we are working on the continuous manufacturing, and now I think we have guidelines. So I use this hot melt extrusion without temperature for the continuous manufacturing. So in that case, I use the 3 hoppers. One is for the drug and combination of some excipients, and then the polymer, and then the excipients for the lubrication. So that was the steps until they get the final granulation. So it's possible that you don't need to use the temperature. But if you want to use the temperature, yes, there are some excipients that Carnauba wax. I developed the modified release formulation for using the Carnauba wax. So by 35 or 40 degrees Celsius, you can melt that. So for the granulation for this, so that's possible. So it's up to you that if you want to use 35 degrees Celsius, it's possible. If you don't want to use the temperature, that is also possible.
Okay. And the other question is just more a curiosity. You mentioned, and it's, I also agree with you that eventually, say by 2030, you can have 3D printing in your house and print your own tablet. The big question is, my opinion, how can you handle the QC, the quality control of when you print your tablet in your house?
Yeah.
I know of course, it can have safety issues for patients. We'll get there, I'm sure, but the question is how.
So as initially that most of the things that we was working on the continuous manufacturing, that how it will be. So like in 10 years before, when I started working on 10 years before of this continuous manufacturing, people were asking how we will manage this. How will be like the analysis of we will done. So the same question now the people are asking for 3D printing, that how we'll print at home. So that's a real logical question. So that it will taken care by the team. And of course the guidelines are also coming now. I think they are on the draft stage. And they are taking care of the quality control, that how we'll manage the quality control. So here that will put that when the doctor will prescribe. So additionally, when you go to the pharmacy, because it will not available anywhere. So it will be obviously available with the prescription. So he will, at present, the plan is to to manufacture it at the pharmacy level. And the person who is handling that, he is already pharmacist. So he knows how to take care of this. So the regulatory agency will give the training to them, and they will take care of the quality part. But afterwards, if it is successful, then the regulatory agencies are planning to plant it at their personalized level, like maybe at home. So now we are presenting that they are giving some trainings to the pharmacists who will print this. And based on this successful like pilot plan, then definitely we'll move forward for the next stage.
Thank you very much. Any questions in the room? Okay, I'm gonna ask you a bit of let's see the question. You talked to us about the goodness of amorphous solid dispersions. Okay, what are the drawbacks?
The first one is the stability. That's the biggest drawback of the solid dispersion, because when you convert from the crystalline to the amorphous form, there are some molecules, they are getting precipitated out, maybe during the humidity or at high temperature. At 2560, mostly they should be stable, but when you go for the higher temperature, 3075 or 4075, they are getting precipitated out, or they are converting in the crystalline form. So that's the stability issue is the biggest problem. Or polymorphic formations. So there are different polymorph forms. So these are the two biggest issues some I face. And sometimes even after 3 months, solubility issues is the biggest problem. So like initially they show that the solubility enhanced 100 times, but they are decreasing in some due course of time. So these are the few challenges or few drawbacks of solid dispersion. But of course, there are remedies that we can overcome with the proper knowledge and proper selection of the excipients for process. So yeah, it's possible. But of course, every technology has the drawbacks.
Very good. What about when you are studying the stability of the dispersions that we look at, and when you got to look at the kinetics of the relaxation, looking at how the glass transition changes over time, and the kinetics of the crystallization, see whether a new form or a new crystalline form is appearing over time. How do you take into consideration the loading? Because obviously you're going to make more difficult the analysis by having more components in the system, and the amount of components is also very important. How do you take that?
That's what I think most when you go for the formulation development, the solid characterization technology is a very useful tool. Without that, I think no one can develop the formulation, the robust formulation. So take the advantage of this solid state characterization technology like DSC, or advancement of this new technology, like along with the AI, XRD, DSC, or Raman spectroscopy. Use the PAT tools if you are using the continuous manufacturing. With this integration of this new technology, you can develop the robust formulation. I hope I give the answer to your question.
Okay, any more questions? I got a final question, probably a rhetorical question. Why is it so much reluctance sometimes to try new excipients? You mentioned some companies doing novel excipients, but why there is such reluctance to try something new? I very rarely come across anything that hasn't been used for 25, 40 years already.
Actually, there are excipients which are available, but some problems are that they are not suitable for each and every API. There are different APIs, so sometimes we need to try the different API with the different excipients. So there should be one API that should be useful for each and every API for BCS Class and BCS Class 4. That's the reason I think it should be stable. It should have providing the good solubility enhancement, giving a good amorphability. So we need some unique polymer, so that, and also the low melting temperature and good processability. So that's what we're looking for. I'm not saying that, but still I think it's better to develop some new polymers.
Very good, thank you. Any final question? Okay, thank you very much, Devendra.
Thank you everyone.